All right, so welcome again, everyone. Thank you for joining us right in the middle of the summer where everyone is vacationing. Today, we're likely to have Dr. Israel Borokini here with us to talk to us about his research. Dr. Borokini completed his PhD in ecology, evolution, and conservation biology right here at UNR. Before starting his PhD, he's done several research studies in Nigeria, his home country, and he has published over 50 technical papers. He's a member of several professional societies. He holds leadership position in several of them, including president of the Africa section of the Society of Conservation Biology and director at large for the Northern California botanists. Dr. Borokini actually will be starting in a couple of days an exciting research postdoctoral fellowship at the Department of Integrated Biology at the University of California, Berkeley. So we're glad to have him here with us to tell us about all the research that he's done over his PhD before leaving to UC Berkeley. Dr. Borokini, the floor is yours. Thanks for joining us again.
Thank you very much, Maha, for that introduction. And I appreciate everyone for taking your time to come over to listen to my talk. And also want to recognize the presence of one of my advisors. So we work together on this study, Dr. Kenneth Newser from Geography Department, who is also listening. This discussion or this presentation is one of the chapters of my research, which is perhaps the most exciting of them all. And this is where you did a lot of fitting of niche models for a particular federally threatened species that is local within this region. I'm happy to present on the results or the findings of that study. All right, so just by way of introduction, I'm hopefully going to try to explain this in layman's terms. So ecological niche models is basically a way of describing the functional or the environmental niche or position of a particular species in the environment. So there are different ways of describing the niche or ecological niche of a species. Some describe it in terms of what are the range of conditions that support the persistence of that particular species. Some what is the range of the tolerance, ecological tolerance that the species can, under which the species can survive. Other experts describe ecological niche in terms of the position of that species in the ecosystem. Is it a predator? Is it herbivore? Is it a green plant? Is it a producer and things like that? So, but in terms of ecological niche modeling, in terms of the concept of the modeling or the fitting of models for ecological niche, we describe it in terms of the environmental niche, that is the range of conditions under which the species can survive. And knowing fully well that not all that species do not fully occupy the entire range of conditions under which they survive, then we have what is called the potential niche. Then we have the realized niche. And the realized niche is like where we actually found them. And then we have then we have the potential niche, which are areas where they can actually live, but they not yet have the opportunity of reaching those areas. So we fit niche models using environmental conditions where we know we have found a species, and we use it to predict to other areas where they have not yet reached, but we think that the models can predict that they can actually survive there. And in literature, there are several studies people call niche models species division modeling. Some call them habitat models. Some call them bioclimatic envelopes or climatic envelopes, and particularly, especially if you are using only climatic variables for that modeling, then it's more appropriate to call it bioclimatic envelopes or bioclimatic models. And in most cases, we use the occurrence points or areas, the spatial data, oftentimes the longitude and latitude, that is the geographic coordinates of where the species have been found, and the areas where they have been predicted that where they has been surveyed, and we know for sure that the species is not there, Or in many cases where we don't have those kind of information, which we call true absence points. If we don't have those, then we use background points, which are called pseudo absence points. And then we use those as dependent variable. And then predictors, this is where we use environmental variables like maybe precipitation is important for this species. Maybe minimum temperature is important for this species based on existing scientific understanding on this species. And in many cases where we don't have this kind of information to know which variables are important for this species, people just use a bunch of different models, environmental variables, and hope that some of them are important, that the models will flag some of them to be important at the end. And in recent times, there have been studies that now use functional traits, like like different important morphometric or anatomical variables or information on that particular species to see whether it correlates with important environmental variables. And some use genetic clusters depending on what kind of question you are asking or the investigation. And niche modeling has a lot of applications as important because we can use niche models to actually identify which predictor variables have ecological or biological importance to that species. For example, let's say a particular species, species A, we fitted the models and we found out that temperature was really important. And then we can use that to make predictions that's based on the fact that temperature was important. If global warming continues or if climate change continues, then the niche of that species is going to shrink because areas that used to be suitable or favorable for this species may no longer be available. So we can use those, we can identify important variables, and then we can use those to predict what the future will look like or downcast or backcast how the species distribution used to look like in many, many centuries back. We can also generate habitat suitability maps, and we can use that to estimate the species range, how rare is the habitat for the species, and we can use it to guide field surveys to new areas which were predicted to be suitable, and to go and find out whether the species is in fact found in those areas, which is where I'm going to be presenting on. And in literature, niche models are being used to discover new populations of species, new species completely, and even some species that have been thought to have gone into extinction. Niche models have been used to discover, to find new populations. And also it could be used for future planning, like areas which are predicted to be suitable for a particular species, but the species is not there. Those areas could be earmarked for future experiments or translocations or for corridors and several other conservation planning. And you can also use niche models to find out whether evolution between two sister taxa, whether it is allopatric or sympatric, that is, does the evolution of two species, does it involve a complete change in the environmental conditions or the niche for species A from species B? So we use those kind of information to make some predictions on certain evolutionary processes that involved in the speciation of two species that are closely related to each other. And that is really, really important, especially in the Great Basin Desert, where there are so many species that are highly related to each other, and they have a lot of habitat specializations. So, but the problem with niche modeling is that it has because a lot of conservation effort is done on range restricted taxa, because when a species has a very small geographical distribution, they are more vulnerable to extinction. They are more vulnerable to several things, environmental or genetic processes that can result in their extinction. So most of the species that are listed in Endangered Species Act for the United States, or the IUCN Red List of Threatened Species and so on, most of these species are species that are highly threatened or they have a small range. And the problem is for rare species is that they have few points. And when they have few occurrence points, many models or the algorithms that we use for fitting these models, they are very sensitive to small data set, and so it could lead to over prediction, and sometimes also biased prediction. And oftentimes, because they are rare species, many of them have not been well studied, especially Ivesia webberi that I worked on. Almost nothing is known about this species until I started my PhD. So I started from the scratch, making a lot of assumptions on what I think are important variables for this species. And the same thing for many other rare species out there. And so in this study, what I did was a 5 years, multi-year, 5-year studies of iterative niche modeling and field surveys to find out what are important ecological variables for this species and how, and then when I fitted those models and I go out to the field to go and find out, to go and survey areas that are projected to be suitable, to go and find out whether you can find the species there. And that resulted in the discovery of many new populations for this species. And then I had questions that between the original populations or data I started with and the new data set that I discovered from the new locations, what is the degree of the similarity between the ecological conditions between those two sets of data? And that is what the second question is focusing on. And so this is basically, this is an illustration of what the 5 years of work is that I have data set, I fitted some niche models, and then I use that models to generate habitat suitability maps. And then I took those maps to guide field surveys, driving all over the place, looking into projected suitable habitat to see whether the species are there. And areas where it is predicted to be suitable, but we didn't find any species there, we took data from there and we added them as absence points. And where we found new data sets, we also included them as well. So, and then with additional data sets, we put them into the original data set and rerun the models again. So we keep tuning the models with new additional data set, and until we feel like, well, it is over, it is done. Not that the work is done, but you are done with your PhD, you need to get out and move forward. So basically, and so this is what we did. This is the framework for the niche model. But more specifically, so we have presence points and absence points, and then we have a lot of absence points. We thin them, and then because we don't know which variables are important, so I assembled 72 predictor variables, and then we did some future selection, and which resulted in 6 important variables that are also uncorrelated with each other. And then, and by the way, we started with 23 spatial presence points. So we have only 23 presence points when we started, and about 758 absence points. And so we could only use 3 predictors at a time because we have less than, because there have been some recommendations that use 1 predictor per every 10 occurrence points. So because we don't have a lot of occurrence points, we could only use few predictors in the modeling. And the modeling, we did cross-validation, 80% subset, and use 6 different algorithms, and the habitat suitability models or maps were generated using a weight mean ensemble in BIOMOD2 R package, using TSS as the metric for model performance evaluations. Yeah, and then it is the habitat models or the maps or the habitat maps that were used to guide field surveys. So how did we do the model or the field sampling is that all of the absence points were overlaid on the map, on the suitable map, and then areas that were predicted to be suitable but have not yet been sampled were selected for sampling. And of course, because very few people were involved in it, I was basically the only one doing all of the field work. all of the field sampling. So it took many years to be able to comb many of these areas. And some of the areas have not even yet been combed yet or checked yet. Some of them are found in private properties which are inaccessible. So as more data is being done through field sampling, they were added into the models and rerun and then did further sampling. And so we discovered 9 new populations, which increased the total occurrences from 23 to 32. And the northern species range was extended by 65 kilometers because, as I will show in the results. So from the results, we discovered that across 2015 to 2020, which are the 4 different years that I did the modeling iterations, we discovered that the perennial herbaceous layer, which describes the areas that have a higher, which has a higher spatial distribution of forbs and grasses, native grasses and forbs within the region, as opposed to shrubs and trees and other areas. So the areas, the spatial area map that shows the perennial herbaceous plants was shown to have the most important contribution to the models, followed by the topographic position index. And this topographic position index shows it's a kind of a neighborhood-based elevation topographic variable, which shows compared to neighboring cells, what is the elevation of a particular grid cell compared to other neighboring cells. So it's a kind of a topographic variable, which is a very important variable, given that the Great Basin Desert and the region where we have, for many of these plants, there's a lot of topographic complexity, which results in a lot of variations across the landscape. So the perennial herbaceous layer and the topographic position index were the most important consistently, how to perform other variables, and the third variables were across the different years were different. But at the very end, we found that summer precipitation was really, really important as well. And now we generated the residual, the partial response plots for each of these variables, and we found out that the probability of finding Ivesia webberi increases in sites that have greater than 20% perennial herbaceous cover, in sites that have at very minimum 25 millimeters of precipitation, that receive 25 millimeters of summer precipitation, and in sites that have gentle slopes or gentle ridges. So those are the areas where we found that are suitable for this species. And these are some of the pictures that shows what we're talking about. So this plant, these ones where we have the cursor with these tiny, smaller yellow leaves, that is the Ivesia webberi, and the one with the bigger yellow leaves, that is Balsamorhiza oakeri, which is another species that is found in association or in close, almost all of the sites where this species, Ivesia webberi is found, we always find these other Balsamorhiza species there. So don't get distracted by that one. It is the smaller plant that has tiny yellow leaves. That is the one we're referring to here. So you can see that this site has a lot of perennial herbaceous plants, including Ivesia webberi. There are some parts of it that is rocky, that has rock on them. There are some other parts that have just bare ground microsites. The same thing here. This is another example of the sites where you see a lot of Balsamorhiza oakeri there, and then you see Ivesia webberi also found within there. But you can see that in this site, most of the plants have completed their flowering season, and that is one of the problems we have with this Ivesia webberi, because I can only do the field sampling within a 1-month window period in a year. If I miss that 1 month, I miss 1 full year of studies of field sampling, because the best time you can find this species, especially in an area where it has never been sampled before, is when this plant is flowering, when detection probability is very high. So, and then to answer the second question, to find out whether the environmental conditions in the original occurrence points, whether they have degree of similarity with the new locations, I put them in, there are two ways of solving this problem, but in literature, what has been proposed is to do an ordination-based analysis. So the first thing I did was to plot an ordination with the 6 uncorrelated important predictors using all of the presence and absence points, and then extracting the PCA scores for the occurrence points, for the presence locations, both the new presence locations and the original presence locations, and then use those to estimate the degree of a similarity in the environmental conditions in the new points versus in the old point. But before I go into the results for that one, the plot itself, the biplot, the PCA biplot itself shows that topographic variables and climatic variables are really, really important for this species. And just this is basically what we discovered here as well, that climatic variables and topographic variables were really, really important from the variable contributions, which is what the PCA also gave us. So what we did here, so there are several metrics. There are fairly new metrics that were developed, which helps us to estimate the degree of similarity or the degree of overlap between two spatial data sets. In fact, this method was developed for estimating the degree of similarity between a plant and invasive species in its native origin and in its introduced origin. For example, think of cheatgrass, which is a native, which is an invasive species here, but it came from Eurasia. So we can take spatial data sets and the environmental conditions in Eurasia and use it to, and also the spatial data sets from its invaded region in this part of the world, and see whether there's degree of similarity in the environmental conditions here versus in its native origin. If the degree of similarity is high, then we'll say that the niche has not changed that much. But if the environmental conditions are so different, or they are so apart from each other from its invaded region versus its native region, then we'll say that there is a change in its niche. That is, there is likely expansion in its niche. So bringing that kind of an analogy into this situation, we ask the question whether the new points that we have, whether the environmental conditions are slightly different from the original points. And we found out that there was 44% niche overlap, that is the degree of similarity. We found stability of 93, over 93% stability, that is little change in the niche, and only 6.8% expansion in the niche of this species when comparing the old and the new points together. That is, the new points that were added, they did increase or expand the environmental conditions under which this species can survive. And we found only 6.8% expansion and a great overall 93% stability. That is very little change, slight change in the niche. And also using what is called the niche equivalency. So the niche equivalency is another metric that was developed which estimates the degree to which you can use the new points to exactly predict the old points. That is, if we use the new points as training and use the old points as test, can they predict each other with high degree of similarity? If so, then we'll say there is equivalency in the niche. And when we found that, and then you can also do some simulations or randomizations to find out the degree of confidence with which we can accept or reject that niche equivalency results. And in this case, we realized that the p-value that it gave us was greater than 0.05. And what this means is that there is a lot of ecological similarity, but the differences between the new points and the old points does not make the two data sets to be able to reliably predict each other, which means that there is some differences, there is some amount of differences or ecological differences between the conditions in the new points and the old points. And another way of looking at it is what is called the niche similarity, which finds out the degree of differences. Is a little bit counterintuitive because they call the metric niche similarity, but it's actually estimating the degree of differences between the two conditions. And in this case, because the p-value was also greater than 0.05, it shows that that there is some level of dissimilarity in which is the actual word to use. There is dissimilarity between the two niches. So which means that the new points that we discovered, they have some ecological values to them. That is, we got some new information from them that will help us to be able to reliably estimate the niche of this species. Assuming there was no differences between the old and the new points, then we'll say that, well, we've not really learned anything new because the old points we have gave us reliable information and the new points added no new information. But in this case, we found out that the new points that were discovered from these field surveys and these model guided field surveys have some important ecological information which were of value to the estimation of the niche of the species. So let's look at the map visually. Here we'll see the model, the habitat suitability map that was produced for 2015 model. And the green points are the original points and the yellow ones are the new locations that were discovered. So we can see that when we started, we only have these few points, but you can see if you look at the top corner, the top right corner of the first or both maps, you will see two new locations there, which is why we estimated that there was a 65 kilometers expansion in the range, in the geographical distribution of this plant that is considered to be rare and range restricted. We found some new locations of this species beyond what was estimated to be the range of that species. So we found a lot of interesting information from the modeling of the species. And from 2015 to 2020, the number of suitable cells or the cells within those maps, each of the grid cells that were considered suitable decreased by almost 50%, which means that the new information we added helps to reduce overprediction of the niche of the species. And so this is, given the time that was allotted to me, so we've just come to a conclusion from this study that in many cases, especially in literature, many scientists or authors, they just fit a niche model, just one time niche model for a species, and then they write the manuscript and they publish the paper and that is it. But here we argue that just one time fitting of niche models is not enough, especially if you are working with a rare plant or rare species or a range restricted species, because you may not have enough information to be able to reliably describe the ecological niche of the species, but rather use the information you have to fit some initial models and go out to the field, do some field work, and any new data you get from that, introduce it into the models and rerun it to be able to reliably describe the ecological niche of this species. And that is what we have done patiently over the last 5 years. And we discovered that topoclimatic variables are really, really important for this species, and biotic variables represented by the perennial herbaceous layer and the topographic position index. And we also, and another important thing to bring out from here is that areas that are predicted to be suitable, but you didn't find a species there, it may not necessarily mean that that area is not suitable for the species, but the species is rare for a reason. Maybe it doesn't have enough dispersal capacity to be able to reach those new areas. So those areas that were predicted to be suitable, but the species has not been found there, are often called areas of commission errors, but they are in fact areas of potential niche that should be earmarked as potential corridors, landscape corridors for the species, especially as the climate continues to warm into the 2100 and beyond, so that the species have opportunities to be able to disperse across the landscape to their new homes, what their new homes will look like. And also, of course, as I've shown from this study and from this presentation, that the additional occurrence locations for this species are slightly, roughly 7% expansion in the environmental niche space for this species. And, but the environmental conditions fairly remains the same between the original and the new points. And I want to appreciate again Dr. Ken Newser, who is here, who guided me a lot in this study. And also want to acknowledge all my different funding institutions that supported this work across along the way. So thank you very much for your time, and I will take questions now.
Thank you so much, Dr. Borokini, for the interesting talk. We're going to open the floor to questions now. If anyone in the audience has questions, please feel free to just unmute yourself, or you can also put the questions in the chat or raise your hand.